and stop at the time. So what you see here, this is the blood vessel, of course, and you have now here, okay, let's move that, we done this. So you have a cytotoxic T lymphocyte, it's coming from the lymph node, it has been instructed by an antigen presenting cells already activated because it's cytotoxic T cells, so it was a naive CD8, it has become a cytotoxic T lymphocyte, or CD8, it was CD8, cytotoxic, by the help of a Th1 response, and is looking now for what? Is looking for the virally infected cells. How does it do it? And I will show you. Okay, now at the cell surface, this is the TCR we've shown. This is the T cell receptor. There's the CD3 complex, you don't see it, and there should be an MHC class CD8 molecule that is specific to the MHC class 1. Remember CD8, and it's trying to sense the MHC class 1 protein. And as you see in these cells, as I said before, the MHC class 1 present not only the viral peptide, but also the self antigen or self peptide. Remember, and in this particular case, the T cell receptor will sense that this is a self peptide, and he's been instructed in the thymus not to react against self antigen. So basically, what he's going to do, he's not going to do anything. What he's going to do, he's going to see this is a self antigen. This is something I've been taught to recognize. I'm not going to react. Okay, he's moving away. Now this is a virally infected cells. So what will happen is virally infected cells. Let's move a bit forward. Okay, virally infected. What happened is virally infected cell will present not only self peptide, but they will also present viral peptide. Remember, this viral peptide present in the cytosol, processed via the MHC class 1. The MHC class 1 goes at the cell surface, and here is a viral peptide presented by the MHC class 1. Okay, and here it is. And this, how it look, I mean, I'm always amazed. The more I saw it, the more I'm amazed of it. And look what happened. Now the TCR, T cell receptor, recognize that this is not a self antigen. This is a viral peptide, and that there's the virus inside the cells. And what is going to do? This is going to give the signal for the cytotoxic T lymphocyte to be activated. And what will happen? Activation. Okay, to launch a number of cytokines. Say, look, I found an intruder in these cells. You need to come and help me. So more cytotoxic T lymphocyte will show up. But at the same time, this is how it's going to kill the cells. Two phases. The first phase release two sets of protein. Okay. The first protein is called perforin. These are proteins that are assembled together to punch a hole at the cell surface of the virally infected cells. Here, okay? Look how specific it is. I mean, and this is what we call the perforin pore. And then what comes, you have this granzyme. This granzyme here is an enzyme that will drive apoptosis or cell death of the virally infected cells, so that the virus cannot replicate. And therefore, if the cell is dead, the virus will die as well. And what will happen? Granzyme gets into the genomic, inside the DNA and kill the cell. You see how beautiful and how specific it is? No bystanding effect. You know that only virally infected cell will be destroyed by the cytotoxic T lymphocyte. Okay, let's move on. Okay, so yes, so these are all the information so far. So what we've learned, you've learned how the T cells, the antigen presenting cells, activate the naive T cells via by activating the TCR complex, and this via they have each their TCR, except that the difference they have a CD4 molecule to recognize MHC class 1 and class 2, and CD8 molecule that recognize MHC class 1. Now let's see how the Th1 cells will instruct the B cells which antibody to produce. Remember, we have different classes of antibody. We have IgM, we have IgG, we have IgA, and IgG. So the T cells need to instruct the B cell to produce the right antibody. Otherwise, if you don't have the right antibody, you're done. And we're also going to see how the T cells sense the microbe, and how is the sensing of the microbe important for helping the T cells to provide the best signal. It's always. So this is like it's very easy to remember the antigen receptor. On T cells is TCR, or T cell receptor, and B cell is B cell receptor, or BCR. Just remember. But the difference is that the BCR on B cell is an antibody. Okay? Has also the same thing. Each antibody specificity is unique, meaning that every B cells has a different specificity, meaning that they can recognize different microbe or different portion of the microbe, okay? And has two regions, a constant region, a variable region, and you will understand the variable region is there for what? Variable region means that to accommodate a variety of different microbes, okay? That's very important. So, and it's the same process to generate a variety of antigen receptor is the same thing. Is gene segment assembled in a random manner to create a protein that will be able to accommodate different microbe. And the number of combination that you could lead to this antigen present to this BCR is 10 to the 11th, which is a lot. I mean, there shouldn't be any microbe your B cell shouldn't be recognizing. And this is indeed really spectacular. And finally, the difference now, there's a big difference. Not only the BCR is an antibody, but also can recognize the peptide that has not been processed. And it can recognize the whole microbe. That's the beauty of this B cell receptor. It can also recognize small chemicals. So that's the main difference. And I will summarize that. So this is your microbe. The whole microbe doesn't need to be processed for the B cells to recognize it. And this is two BCR, B cell receptor together. And what will happen? You have a cross-linking of these two B cell receptor together, and that will trigger the first signal. Remember, like T cell receptor, you need two signals to activate the B cells. And you will see the second signal, what the second signal is. So the first thing is the microbe doesn't need to be processed. B cells sense it and then gets activated. So that's the one. And at the same time, B cells, remember, this is an antigen presenting cell. So in order to fully activate B cells, they need the second stimulatory signal, which is B cells. And I will show you what it does. Anyway, at the same time, what happened is that the cells get activated. This microbe gets inside the cells, processed, because remember, it's an antigen presenting cells as well, in addition to producing antibody. This microbe processed in different peptide will be expressed by the MHC class 2. That's what it does. It can't process via the MHC class 1. And what happened is, here is your microbe that was recognized by the BCR, that has been cleaved, that has been processed via the endogenous pathway, expressed by the MHC class 2, expressing this B7 protein, which is very important for T cell activation. Here comes T helper cells that has been already activated by the antigen presenting cell. So the TCR, the T helper cell, which is CD4, knows what microbe is looking for, because it's been instructed by the antigen presenting cells to look for a specific microbe. And only when you find the B cells with the right microbe that you will be able to fully engage. Okay? If this is not the right peptide for this TCR, the TCR won't get engaged. It's only when there's the right match between microbial peptide and TCR that you get activation of the helper T cells. And look what will happen. This is very important for you to understand. So you have the two necessary signals, TCR, MHC, CD28, B7 on helper T cells. You have the two important signal, T cells will get activated. And when it gets activated, there's a new protein here called CD40. And this 40 is very important. Why? Because this protein here, CD4 ligand, very important. I usually like to ask the question at the exam, so write it down. Is because of this protein, we can switch the type of antibody the B cell will produce. Remember, when you were first exposed to, let's say, we don't hope, but let's say you were exposed to this COVID-19, then the first antibody you're going to produce is IgM. However, the best defense you want is IgG, because IgG is the best immunoglobulin that you can have. It can opsonalize the microbe and activate complement, while IgM can only activate complement. I'm going to summarize that at the end. And in order for the B cells to switch the type of antibody we produce from IgM to IgG or IgA or IgE, we need this interaction between CD4 ligand and CD40. Why this is important? Because you have a group of patients who do not have a mutation in this protein here, CD40 ligand. And they give you a syndrome called hyper IgM syndrome. Why? Because the only antibody that the B cell can produce without the CD40 ligand is IgM. And therefore, in response to any infection, the patient will only be able to produce IgM, because it doesn't have the signal on T cells that will tell the B cell to switch from IgM to IgG. Okay, this is called hyper IgM syndrome. This is something that, if you want to write it down for you and read about it, this very, very important patient coming with recurrent persistent infection. We're going to cover that in the immunodeficiency. So, but it's very important for you to understand that the importance of T cell in switching the antibody. And this is what I said. Once you have all this, then the cells can proliferate, differentiate, and produce the right antibody against the infection. Question was also for you at the exam. Usually I would show you this, and I will tell you, and I will ask you to tell me what is 1, what is 2, what is 3, what is 4, what is 5, and 6. And here's the answer here. So please make sure you know about it. Okay. I would find it unacceptable for immunology student, student studying immunology, not to understand the basic of an immunoglobulin, especially now that we are using it to treat an array of different diseases successfully as a form of monoclonal antibody. Anyway, so remember what we're going to say for the clinical case together. So the first antibody that is produced that doesn't require T cells is IgM. So whatever you're exposed to, Streptococcus pneumoniae, Staphylococcus aureus, flu virus, you're going to produce IgM. However, you're going to have IgE or IgA or IgE depending on where the infection is, and more importantly, with the help of T helper cells. And more importantly, the interaction between CD4 ligand on T cell and CD40. Okay, very important. And what is beautiful about T cell antibody production is that the more exposure to the microbe, the greater the affinity is, meaning that the antibody that the B cell will produce will bind with the greater affinity, meaning being able to detect very low amount of the microbes. I mean, it's just, I'm just always amazed by this. And obviously, what makes us alive today is the immunological memories. And therefore, when the B cell is in contact with the microbe, there will always be memory B cell that will remember the microbe, so that next time it doesn't need anything, it will just produce the right antibodies upon exposure with the antigen. So memory is why vaccine are so efficient. Memory is why we are alive today, and we can fight exposure against different microbes. Okay, so this is really summarized. Let's say you are exposed to the influenza virus. The first antibody you're going to make is IgM. As you can see, this is 5 different immunoglobulin associated together. This is thymus independent, meaning T cell independent. And this is classical. It's very powerful because it can activate complement. This is a massive complement. But it doesn't do this. That's the only thing it does is buy us time, activate complement until we can produce a factor of the adaptive immunity. And what happened later? You have T cells. T cells, remember, they've been already primed by antigen-presenting cells. They make contact with the B cells, and by the CD4 ligand and on T cells and CD40 on B cells, the T cell will instruct, depending on the nature of the microbe, to produce the right antibody. And the best antibody response we have is IgG. In case of worms, parasite infection, the antibody would be IgE. And at the mucosal surfaces, you don't want IgG, you want IgA. And that's what we have at the mucosal surfaces. So that's what happened. And